Okay. We're going to look today real quickly at the structure of a what?
Chromosome.
Chromosome. What does structure mean?
Shape.
Good. Shape. How it looks. Structure of a chromosome. Chromosome is basically condensed what?
Chromatin.
Chromatin, which is
DNA.
DNA. Okay. So DNA is in the shape of a double helix. So I'm just going to start out. I'm going to draw me a little bit of DNA in that double helix shape. Here's my DNA model right here. So here's our DNA double helix, in case you have forgotten what it looks like. DNA double helix, it's got 4 bases: adenine, guanine, cytosine, thymine. Correct. DNA is going to wrap up around proteins. Does anybody remember what those proteins are called? Yes, you need to write this down. They started with an H. Histones. So it wraps up around proteins called histones. And so we get these kind of DNA kind of wrapped up and coiled up around these histones, like so. And then those things wrap around each other, and they wrap around each other, and they coil up into, finally, a chromosome, which is going to look like this right here. Makes that nice X shape. I'm drawing it like this because I want you to understand that chromatin is the loose form of DNA, where it's just DNA wrapped around those proteins, those histones. Of course, we said this shape was what? What's the shape of DNA? Double helix. Double helix, y'all.
My friend.
His name is DNA. His name is DNA. So, DNA wraps up around the histones, and that makes chromatin, and then the chromatin wraps up into chromosomes. So I'm going to label this as a chromosome. Now the chromosome has some parts, and we're going to talk about those parts. Before the cell divides, what do we have to do to the DNA? We have to make a copy of it. And we said the reason is because, everybody put your hands like this, make the big cell. This is like one cell, right? Everybody got this one cell? Don't be shy, Evan. This is your cell. You got cells. Everybody's got them. Don't be ashamed. And your cells are going to divide and become 2 cells. So one cell, in mitosis, 1 becomes 2. So if we only have 1 copy of DNA, not each cell is going to get a copy, right? So we have to copy the DNA. So we have a set for each cell. So this chromosome represents both copies. We call this a sister chromatid, and it represents 1 copy of DNA. This whole thing right here, this whole side, that's 1 copy of the DNA. So what do you think the other side represents?
The other side.
The other copy of DNA. So this other side is also called a sister chromatid.
Chromatid?
Chromatid with a D. Chromatid. You heard it first here from Mr. Hall. And it represents the other copy of DNA. Now, that's why, what's going to happen to these in mitosis? They're going to split apart. And so 1 copy of DNA will go to 1 cell, and the other copy will go to the other cell. Thank you for your enthusiasm. Now, the only other part of a chromosome that we want to look at is this center where they're connected. Let me do this in green here. This center where they're connected at, it has a specific name. We call it the centromere. Centro makes you think of what? Center or central, and it is the center part where they're connected.
Does that come from like the centrioles or whatever?
No. Don't get those confused. Centromere, M-E-R-E, centromere. And this is actually just, if you wanted to define it, you could say the central part of the chromosome that holds together the two sister what? Chromatids. Central part of chromosome that holds the chromatids together. Boom. Now we're going to look real quickly at how this applies to mitosis, and this is where the name and the stuff can sometimes get a little confusing, but we're going to try to dispel the confusion. That's the goal of my life. If you're confused about anything, I want to correct that. Okay, here we go. So I'm going to draw a phase of mitosis. You tell me what phase it is. Is this a plant or an animal cell? Probably. Animal cell. If we're all lined up in the middle, what phase is that? All right, so that's metaphase. And this thing right here, now notice, whenever I draw my chromosomes, I don't draw them like this. And there's a reason for that. I always draw them like this because I'm drawing them as two sister what? Chromatids, because eventually those are going to be torn apart. So, right now, what's this thing coming out and grabbing these? Spindle. This spindle fiber right here is attaching to this sister chromatid on the left. This one is attached to the one on the right, and they're going to pull them apart. So, this represents two copies of DNA. One copy is going to go to the left side. The other copy is going to go to the right side. And we call this a chromosome. And a chromosome is made up of two, what? Sister chromatids. Now, don't let this part confuse you. What phase comes after metaphase? Anaphase. And what happens to those chromosomes? They split apart. As soon as they split apart, we don't call them sister chromatids anymore. We call them chromosomes again. So this right here is also called a chromosome. When these two chromosomes are stuck together, we call them chromatids. But as soon as they get ripped apart and start going to the different cells, we call them chromosomes again. So that may be, I think, the only point where we might get confused on naming them. Let me run through that one more time. When they're together, we call them sister chromatids. When we rip them apart, again, we call them chromosomes. And they each represent a copy of what?
DNA.
A copy of DNA. How many chromosomes did we say humans have?
46.
46. So, if this were a human cell, we'd have 46 chromosomes lined up in the middle. How many sister chromatids would we have? 92. We'd have twice as many. And then in anaphase, when they get ripped apart, we'd have 46 chromosomes going to one cell, 46 chromosomes going to the other cell. So our new cells would have 46 chromosomes each, and that's right for humans, and that's what we want. Okay? All right, this has been our discussion of chromosome parts and functions. Thank you very much.